Hello everyone, my name is Shashika Hevavitarana. I'm one of the plant pathologists at the Cal Poly Strawberry Center. I'm going to talk to you about an industry funded project which is on how soil fumigants affect the soil microbiome over time.
Earlier version of this work was presented at Oleg's meeting this year. What is new in this talk is that we have soil microbiome data now. Let's first understand what soil microbiome is. It includes all the microorganisms in a given habitat. For an example, surrounding strawberry roots, there is a rhizosphere microbiome. In our gut, we have gut microbiome. The microbiome typically affect the health of the host. In this project, we wanted to find out how use of 4 different commercial fumigants affect soil microbiome over time, and if there are organisms that persist and thrive following fumigation. Our aim was to utilize this knowledge in long-term disease management. We measured inoculum levels of Verticillium dahliae over time, plant mortality and yield, and we also assessed soil microbiome changes over time. In this study, our treatments were Ali-33, Dominus, K-PAM, Trichlor, and no treatment control. We conducted the study in our Cal Poly Field 25 Block 4 that were randomized into 6 blocks. Each plot was 150 feet long, and these were our 6 blocks. Soil in this field is clay loam and was very difficult to work with, and you can see the soil triangle here. Our experiment started in June 2019. We bedded up and fumigated in June and planted cultivar Portola in July. We conducted 4 marketable yield analyses in October and November 2019 and twice in April this year. We mowed the plants in January. We didn't see a high disease pressure, so we water stretched plants. The experiment was taken down in May. Yellow arrows here show when we sampled soil. Here's a photo when we planted last July. You can see how hard to form our beds was due to the clay loam soil in this site. You might not believe, despite the condition of our beds, the plants grew really well. Let's look at Verticillium dahliae soil population changes over time. As we know, the threshold of Verticillium that can cause Verticillium wilt in strawberry is 2 colony forming units per gram of soil. This is how the soil population changed over time for all the treatments. We saw that in Ali and KPAM, Verticillium soil population significantly declined at time point 4 compared to its initial level. Despite the Verticillium soil population level above the threshold that requires for disease expression, we saw a minimum level of plant mortality in this trial, which was less than 1%. There was no significant effect on the treatment on plant mortality. This is our marketable yield data. There was no significant difference between treatments again. Our main aim of the project was to look at soil microbiome changes. For that, we sampled soil at 5 sampling points. However, today I am only going to show you the first 4 sampling points. We extracted soil DNA and conducted Illumina sequencing. Basically, you get to see most of the microorganisms such as fungi and bacteria using this method. If we first look at our pathogen, Verticillium dahliae, based on sequencing data, this is how its population changed over time. We looked at many known biocontrol fungi. The good news is that we saw that these fungi amplified after soil fumigation in all the treatments, like in the control treatment. For an example, arbuscular mycorrhizae that belongs to Glomeromycota increased over time. The effect on biocontrol bacteria was not very clear like fungi, but we could see species like Pseudomonas amplifying over time. Again, we have one more time point that we will assess soil microbiome changes at the harvest of plants. However, I don't suspect that the trends would be any different. Take-home messages from this study were that soil inoculum level did not predict verticillium wilt, and it was inconclusive the fumigant effect against verticillium wilt disease incidence. Also, the fumigant effect on yield was inconclusive. However, we found known biocontrol fungi and bacteria increasing over time after fumigation up to the peak fruit production. With that, I would like to thank all our collaborators, the California Strawberry Commission, TriCal, Isagro USA, and AMVAC for funding this project, and our technician Vivian Longacre, and all the students supported this project from the beginning to end. Thank you very much.